not that popular. Small university.
Technician Universität Dresden.
Yeah.
Okay. But this is not what I have written here.
Yeah, so it's basically two affiliations for me. Okay. Okay, so hello everyone. Thanks for the introduction. I'm Florian Kaiser from the Technical University of Dresden in Germany, as well as affiliated to the University of Applied Sciences in Mittweida, which is just close to Dresden, but is rather a small university. And I would like to thank the organizers for giving me the opportunity to present our work here at this great meeting, and I'm really delighted to be here. So in the following minutes, I want to show you how we applied methods from structural bioinformatics, which are developed or were developed in our groups, in order to investigate a very special class of enzymes, the so-called aminoacyl tRNA synthetases. By doing so, we just were able to identify two unique motifs in these enzymes, which we can then relate to the functional mechanisms taking place when interpreting the genetic code. So I want to start off right with the results. Basically, by analyzing 1,000 or almost 1,000 structures deposited in the protein data bank, we were able to highlight two very unique structural motifs in this class of enzymes, which we call backbone brackets and Arginine Tweezers. You will see later why we call them so, because they have some very unique characteristics. And these motifs gives us some insights on how aminoacyl tRNA synthetases work on a functional level, but why is it worthwhile to investigate these enzymes? Well, there's of course one very profound and still open question in biology is, how did the genetic code emerge? And as we all know, proteins are encoded by nucleic acid blueprints, which in turn require proteins for the interpretation, right? So this is somehow a self-referencing system and poses some sort of chicken and egg dilemma. Which were first, proteins or RNA or nucleic acids in general? I don't want to start off a debate here, but I want to focus on a very particular class of enzymes that is at the very proximity of this mechanism. And in order to understand this, I want to show you that the genetic information, which is encoded in nucleic acids, is in turn interpreted by matching codons, so triplets of nucleic acids, to the respective tRNA molecule. And now the aminoacyl tRNA synthetases come into play, because these enzymes are basically the enzymes which covalently attach amino acids to their respective tRNA. So in essence, these enzymes decide which codon goes for which amino acid, and are thus at the interface between gene and gene product. So one can say that these enzymes are the real implementers of the genetic code, because they decide which codon codes for which amino acid. So, just missed one. Aminoacyl tRNA synthetases, or ARS, as I will call them in the following, covalently attach amino acids to tRNA molecules. Well, they do so by binding the tRNA molecule, which is shown in dark blue here. And there are two recognition mechanisms taking place. One is at the codon binding site, which is approximately around here. And the other one is at the amino acid binding site, where the amino acid is fused to the tRNA molecule. So this is basically here. They do attach the amino acid to the tRNA molecule by consuming ATP, and then covalently linking the amino acid to the 3' end of the tRNA. Well, of ARS, there exist two very distinct classes which are dissimilar at all levels we know, at sequence and structure. For each of these classes, we have further 10 types per class, which are responsible for one particular amino acid. So this, in the end, sums up to all 20 amino acids being handled by these enzymes. So for each amino acid, for each respective tRNA, one concrete implementation of these enzymes exists. So if you just look at the classes and the ligands they incorporate, the amino acids, you can just see at the first glance that there's no overall separation, for example, based on physicochemical properties of these ligands or of these amino acids. And also if class 1 amino acids were shown to be slightly bigger in tendency, there's no overall distinction possible just by looking at the ligands they incorporate the amino acids they load to the respective tRNAs. Further, these classes do not share, as I already said, no sequence or structure similarity at all, so they are completely dissimilar. And also at the functional level, in terms of how they bind the ligand, which is shown here on the right, where you can see a ligand of ARS, which consists of ATP part and the amino acid at the end. is almost like a mirror image for the two classes. So even the binding mode of the ligand is completely different for these class of enzymes. And of course, they somehow constitute, again, a self-referencing system because class 1 somehow provides the building blocks for class 2, because of course class 2 is also a protein and consists of amino acids that are handled by class 1 and vice versa. So this class distinction is very unique in several ways. So of course the question arises why there are two classes of ARS. And I do not want to go too much into other theories here, but there are some which can be, for example, that these two classes are of independent origin, so they are not evolutionary related at all. And other hypotheses say that they might have some very ancient common ancestor in the very beginning of life. I want to show you a theory which is somehow a mix between these, because the so-called Rodin and Ono hypothesis, which was postulated in 1995 by Rodin and Ono, states that the two classes emerged concurrently during evolution. So very early at the beginning of life, both classes were there concurrently have been encoded complementary on the same strands of a single gene. This is what they call the so-called Ur-gene, and that simply states that class 1 and class 2 were coded once on a sense and antisense manner in a very ancient form of this coding gene. So nowadays you cannot observe this anymore, but there were some experimental studies that could somehow show that by simply deconstructing contemporary sequences and contemporary structures of ARS, they were able to identify a region of only around 46 peptides, which they call the protozyme region, that still has some catalytic activity in terms of activating the amino acids with ATP molecule, which is a precursor step before the amino acid is attached to the tRNA. So this is very interesting and supports in an experimental way the hypothesis of Rodin and Ono. So how do ARS bind this ligand in the binding site? So if you are looking at the active site architecture of ARS, there are basically two parts. So one, as I already mentioned, is the so-called specificity determining binding region, which is responsible to, or which is necessary to recognize a particular amino acid by forming highly specific interactions with this ligand. So this is to be ensured that not the wrong amino acid is attached to the wrong tRNA molecule. And this part of the binding site differs for all 20 implementations of ARS. On the other hand, there's the so-called ATP binding region, which is somehow the most basal and unifying characteristic between all these types of enzymes, because all of these enzymes require to bind ATP, and also this is the principal energetic barrier in order to fulfill the enzymatic reaction. So in this study I present you, we wanted to investigate how ATP binding takes place in these aminoacyl tRNA synthetases. And we raised the question whether there is a similar recognition mechanism for the ATP molecule in class 1 and class 2 ARS, or if there's another layer of difference between these two classes. So the short answer is no, but you will come to that later. Okay, so how did we do that? So basically we took all structures of ARS which were available in the protein data bank and contained somehow the active site, the catalytic core domain of these enzymes, which was almost like an equally balanced data set between the two classes, class 1 and class 2. And then we applied the protein-ligand interaction profiler, which is developed in our group, to investigate the protein-ligand contacts between the ligand and the binding site residues. So then for class 1, we notified that there were really highly conserved binding patterns in terms of backbone hydrogen bonds with the ATP ligand, while for class 2, there were highly conserved binding patterns between arginine residues and the ATP part of the ligand. So this was apparently there in all class 1 and class 2 structures respectively. And the most stunning thing about these two binding patterns is that they're implemented in a completely different way. So I want to emphasize here that you have to look at the structure to understand the mechanism of class 1 ARS, because this pattern, which is highly conserved in structure and also in function in binding the ATP, is not visible at sequence level at all, because the N-terminal residue does not show any sequence conservation, while the C-terminal residue shows some slight preference for hydrophobic residues. But in the end, you would not have been able to spot this pattern just by looking at the multiple sequence alignment, for example. On the other hand, for class 2, there's completely the opposite taking place. You have two arginine residues which are highly conserved in all class 2 structures that mediate this binding of the ATP ligand. So we published the results in PLOS this year, and I want to get a little bit more into detail how these patterns bind the ATP. So due to their geometric characteristics, we call them backbone brackets and arginine tweezers, these two structural motifs. And the class 1 ARS, they bind basically the ATP ligand by forming so-called backbone hydrogen bonds, which means that the side chains of the residues are not involved into binding the ligand at all. So the interaction is only taking place between the backbone atoms of the two motif residues and the ATP ligand. On the other hand, for class 2, we have this highly conserved pattern of salt bridge interactions and bication interactions. So while the N-terminal residues, and this is important to see here because it refers later to the evolutionary trajectory, the N-terminal residues interact only with the alpha phosphate of the ATP molecule, or the AMP molecule if it's already fused to the amino acid, while the C-terminal residues interact with the adenine and ribose substructure of the ligand. So we wanted to analyze these patterns a little bit further in terms of how they interact or how they mediate their interactions. For this, we applied our structural motif detection methods that are able to dissect the structural motifs at atomic level. Fit3D is the method. And we used this to find out that the Arginine Tweezers pattern uses highly specific side chain interactions to mediate the interaction with the ligand, which differs significantly between the ligand-bound and ligand-free state in our dataset. And on the other hand, the backbone brackets residue shows a strong shift in the alpha carbon distance between the ligand-bound and ligand-free state, which confirms an already experimentally observed, for individual ARS structures observed, rearrangement of class 1 ARS structures in the binding sites upon ligand binding. So we can postulate that this structural rearrangement is a general mechanism that is observable and takes place in all class 1 ARS structures. So how does that refer to the evolutionary origin of these classes? Well, we were able to map these two highly conserved patterns back to the protozyme region, which was postulated by Rodin and Ono. This showed us that the N-terminal residues of both motifs are located right in the protozyme region, so in this supposedly very ancient and very old part of this predecessor urgene, while the C-terminal backbone brackets residue is located close to the protozyme region. This is the so-called urozyme region. It's a more efficient successor of the protozyme, while the C-terminal residue of the Arginine Tweezers is not located anywhere close to this ancient region of these enzymes. So we can postulate that the age of the N-terminal residues is higher than that of the C-terminal residues for both patterns. So just to wrap things up, I showed you two very unique structural patterns in two very unique families of enzymes, which are self-referencing in several ways, and the two binding mechanisms of ATP are implemented in a completely different way. So the conclusions and take-home. To date, this is the largest computational study of ARS structures that was performed, and it's not only about these two binding patterns. We also did some analysis of binding site interactions in general to foster some further analysis in this field. The two motifs we identified clearly delineate class 1 and class 2 ARS at a functional level and add another layer of difference between the class 1 and class 2 ARS beside overall structure and sequence dissimilarity. The Arginine Tweezers residue is highly conserved in sequence, while the backbone brackets are not conserved in sequence at all, but in structure and function, which again highlights that you have to look at the protein structures in order to understand the mechanisms, which you cannot see just by looking at the sequence. The observations we made somehow substantiate the hypothesis of the ancient bidirectional coding of ARS, the Rodin-Ono hypothesis. And in a nutshell, the study showed how we can use methods from structural bioinformatics to link evolution and genetic coding. So the take-home message is that backbone brackets and Arginine Tweezers, due to their highly conserved way, might have ensured efficiency during the very earliest moments of life, during the formation of the genetic code that was catalyzed and mediated by ARS. So what else do we want to do? In future work, we want to investigate, of course, the other side of the medal, because we want to look at the specificity determining binding region by applying same methodologies, and just want to investigate how does the particular ARS enzyme recognize a particular specific amino acid. And of course the question arises if these two motifs we observed here in these enzymes are general mechanisms that can be observed for ligand binding or ATP binding, or if these are very unique for this group of enzymes. So with this, I want to acknowledge all the people who were involved in this work. So mainly my two supervisors, Professor Labudde and Professor Schröder, but also all the members from the group which were involved in doing this research, and of course also the funding agencies for financial support. So thank you very much for your attention, and please step by at my poster if you're interested in further discussions. Thanks.
Thank you very much. I have two quick questions. We still have 4 minutes. The different mechanism of the brackets and the tweezers, have you observed them in other families that bind ATP?
That's a good question. I've already prepared some backup slides exactly for that question. Let's see where they are. Yeah. So, for example, we were able to observe similar mechanisms for this backbone binding mechanism in isocitrate dehydrogenases. And for the Arginine tweezers, we were not able to see that in any other protein family except for paralogs of ARS. So this is basically a paralog of ARS class 2. So it seems to be unique for the Arginine tweezers, but the backbone brackets, yeah, they are somehow probably a general mechanism. So it's worth to look at these backbone hydrogen interactions.
And the last question that I have is, are there clinically observed mutations that lead to disease, and are they unequally distributed between the two types?
Actually, I'm not too sure if they're unequally distributed, but yeah, there are some mutations which lead to, so that lead to severe diseases. But in the end, if you have really like a knockout mutation, you're not going to live at all. So this is very severe.
You would expect that the backbone brackets are much more.
Exactly. So that's what you would expect. But for this particular pattern, there's no experimental data. At least we could not find it that explicitly tries to mutate the residues of the backbone bracket. So not so far. Thank you.
We have time for